Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Conor Liston will present Finding and Fixing Broken Brain Circuits in Depression. The Brain and Behavior Research Foundation is committed to alleviating the suffering caused by mental illness by awarding grants that will lead to advances and breakthroughs in scientific research. The Foundation is the largest private funder of mental health research grants. Since 1987, the Foundation has awarded more than $379 million to fund more than 5,500 grants to over 4,000 scientists around the world. 100% of all donor contributions for research are invested in our grants to scientists who are working to find breakthroughs in disorders such as addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, OCD, PTSD, and schizophrenia. I'm delighted to introduce Dr. Conor Liston. Dr. Liston is Assistant Professor of Neuroscience and Psychiatry at the Weill Cornell Medical College Field Family Brain and Mind Institute. Dr. Liston is also a 2013 recipient of a Foundation Young Investigator Grant and an honorable mention recipient of the 2016 Clearman-Friedman Prize for Exceptional Clinical Research. Today's webinar will begin with Dr. Liston's presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel. Feel free to submit your questions throughout the presentation. Following the presentation, I will ask as many questions as time permits. And now I'm pleased to introduce Dr. Conor Liston. Conor, the floor is yours.
All right. Thank you, Jeff, for inviting me here. I'm really thrilled to be here. Just getting my slides started. So I am, as you mentioned, I'm a neuroscientist and a psychiatrist here at Weill Cornell Medicine, and I'm going to tell you a bit about one of the projects underway in my lab that was made possible through support from the Brain and Behavior Research Foundation. It's really just getting started, but we're very excited about the initial results, and I hope they'll be of interest to some of our listeners today. So these statistics will be familiar to psychiatrists, but I think to a lot of other folks, they're a little surprising. So by some estimates, psychiatric disorders and depression in particular account for 5 of the top 10 leading causes of disability. They're extremely disabling disorders, and they're extremely costly too. We invest a lot of money in the United States in treating mental illness, and there's a lot of money lost due to the disability from people who suffer from mental illnesses. And by one measure, depression is actually the leading cause of disability. And part of the reason for why this is that many of these disorders, depression, substance abuse, bipolar disorder, schizophrenia, they tend to emerge early in life. And although we have good treatments, there's no cure. And so there's this gradually emerging burden of the illness that occurs over the course of a person's life. And one of the big challenges in treating these kinds of mental illness is that it can be difficult. Any patients or family members who are listening right now will be able probably to resonate with this message. It's often difficult to find the right treatment for the right person. And I think it's not controversial to say that part of why it's so challenging to match individual treatments, which often do work well when we find the right match, but it's challenging to match individual treatments to individual patients, partly because of how we diagnose mental illness today. So this, the Diagnostic and Statistical Manual of Mental Disorders, the DSM, this book, which we use for diagnosing mental illness, has been really influential, and it's done a lot of good for people. It's a thing that has kind of seeped into the public lexicon. Lots of people are familiar with it, and it was designed to accomplish a number of goals, and one of them was to provide a language for psychiatrists and other mental health care providers to communicate with each other about the problems that their patients are suffering from. And one of the goals that they wanted to accomplish in doing that was to come up with diagnostic labels that we could all agree on, so that when one psychiatrist or one therapist saw a patient sitting in his office, he would generally agree with the diagnosis given by another psychiatrist or therapist who saw that patient recently. And one of the ways we accomplished that goal was by creating large categories that tend to combine patients with many different problems under a single diagnostic label. And depression is a great example of that. Depression is diagnosed today, a major depressive episode that is diagnosed today when a person suffers from 5 or more of these 9 symptoms. And that means that there's at least 256 unique combinations, unique ways that a person can meet these criteria. And that's not even accounting for the fact that some of these criteria are kind of opposites of one another, like weight loss or weight gain, insomnia or hypersomnia. And I think a lot of psychiatrists and neuroscientists would agree that it's likely that someone who's suffering from weight loss, no appetite, only able to sleep 4 or 5 hours a night, very agitated, very anxious, is probably not suffering from exactly the same biological problem as someone who has gained a lot of weight because they have an increased appetite, they're craving carbohydrates all the time, they're sleeping 19 hours a day, can't get out of bed, and they feel slowed and lethargic and can barely move. They have no problems with anxiety, but they just have no interest in things that they used to enjoy. And those two people sound almost like opposites of one another, and yet we give them the same diagnostic label today. And before I go on to tell you about how we're trying to rethink this approach, I always like to emphasize that I think that the DSM has done a lot of good for people. It's been really critical in ensuring that patients have access to mental health care resources, and like I said before, providing a language for communicating about mental illness. But it was never designed to be a tool for neuroscience research. And I think a lot of scientists today think that it's not very useful for studying the neurobiology of mental illness in a number of ways, and part of that has to do with the fact that there's this big heterogeneity, diversity in the kinds of patients who get these diagnostic labels, and there's no one-to-one correspondence with their kind of underlying biology. So how do we go about rethinking that? We're certainly not the first people to tackle this problem. There's been a lot of interest in thinking about how to subtype depression for many years. And this work has also been really influential. And the way it's typically been done is we've looked at big groups of patients and we've asked what kinds of clinical symptoms tend to co-occur in subsets of those patients. And based on those patterns, we've discovered subtypes of depression, and we've learned a lot from defining these subtypes. Again, some of them, like seasonal depression, is a term that lots of people are familiar with. It's seeped into the public lexicon. Many people know what seasonal depression is. But the fact remains that there's still, with rare exceptions, very few biomarkers, objective measures that we can obtain from patients, either by measuring blood levels of hormones or by obtaining a brain scan or some other biological measure that has a one-to-one correspondence with any of these subtypes, and that's really kind of useful in widespread clinical practice for diagnosing them, or again for matching treatments to individual patients. So our approach in the study I'm about to describe was to kind of turn this upside down and ask whether, instead of starting with clinical symptoms, could we start with neurobiological measures and ask whether we could cluster patients on the basis of kind of co-occurring patterns of neurobiological differences. And then if we could subtype patients in this way, were those subtypes useful for predicting interesting or clinically useful differences in the way their symptoms present and the way that they respond to treatment. And that's basically kind of the rationale, the background for the approach to the study I'm about to describe. But before I can jump into that, that approach begs this other kind of more basic question, which is what kinds of neurobiological signatures should we focus on? And we've known for some time that if you obtain an MRI brain scan like this one in the upper left, if you obtain an MRI brain scan from a patient with depression, the kind of brain scan someone might get if they had a head injury and they went to an emergency room to make sure that there was no bleeding in the brain. If you look at these brain scans, the brains of people with depression look basically normal. There's nothing that stands out in the structure of someone's brain at this level that tells you that they're depressed. But these kinds of images belie this tremendous complexity that lies just beneath the surface. So in each one of these pixels in this image, there are hundreds of thousands, probably millions of brain cells. And these brain cells, neurons we call them, have these fine branch-like extensions that enable them to communicate with other nerve cells. And if you zoom in even further, you can see that these branches, kind of like branches on a tree, have these tiny microscopic protrusions that contain what we call synapses. Synapses are connections between brain cells. And they play a vital role in enabling communication between brain cells. And a lot of research in recent years has focused on the role of synapses in depression and other kinds of mental illness. And although we can't measure synapses, visualize them, or even measure their function directly in the human brain in a non-invasive way, we can use brain imaging to get kind of an indirect proxy for how synapses are functioning in the brain. And if you'll just bear with me for a second, I'll tell you a little bit about that before I jump into telling you about our findings. So the way this works is based on a discovery by two really pioneering scientists, Weill and Mark Weill, about 15, 20 years ago now. They showed that MRI brain scans can be sensitized to patterns of blood oxygen consumption in the brain over time, and that the brain at rest exhibits these kinds of spontaneous fluctuations that you see in this video here. The brain, the images that we're acquiring become brighter and dimmer over time as the activity level of the brain changes. And the critical discovery that Mark Weill and Brock Biswell and others made was that regions of the brain that are strongly connected tend to fluctuate together, and that's what you see here. If we segment the brain into a bunch of regions, and then we extract these like a map of these fluctuations over time, and we compare them, you can see that in two brain regions that are highly connected, they go up and down together. And that kind of makes sense. If this red region is highly connected with this blue region, then when this red region becomes active, the blue region is also going to tend to become active. And that's why they tend to be highly correlated over time. And we can use these measures. Excuse me, there we go. We can use these measures to create maps of how different brain regions are connected, and that's what you see here at the lower right. In this image, warm colors denote brain regions that are highly positively connected, and cool colors denote the opposite, brain regions where when one becomes active, the other becomes less active. And it was these maps that we chose to focus on. And in order to accomplish our goal, that is trying to discover new ways of subtyping patients with depression based on these maps, we knew we would need lots of data. And that's where these people you see, if I can jump ahead to the next slide, there we go. These people you see pictured here, that's where they come in. So I like to highlight their contribution first because it's these folks who really made the work possible. I went around the country basically begging everyone I knew who had data like this, and a lot of people I didn't know who had collected data from patients with depression to share their data with me. And the people you see pictured here are the people who are generous enough to do that or to contribute to the project in some other way. Many of these folks shared their data with us before it was even published and otherwise available, and I'm tremendously grateful to them for making this work possible. Just to give you a little bit of background, most of the work I'm going to present to you is based on this first data set, which was collected from 711 subjects. That's 333 depressed patients. These are actively depressed patients, unipolar depression, who are collected from 5 different institutions, that is universities across the country. And we use this data set to discover the subtypes that I'm about to describe. And then importantly, we also had a second data set comprising 477 subjects, 215 patients with unipolar depression. The remainder of those subjects were healthy control subjects. And we used this second data set to replicate our results. So without further ado, I'll jump into telling you what we found. So as I mentioned before, we began by taking the brain scans that we had that were shared with us by investigators from various sites across the country and parcellating them like you see here. And that basically just means dividing the brain into smaller regions, extracting the MRI brain, what we call a bold signal from each of these regions, and asking how each MRI bold signal from each of these regions, how it's correlated with every other region. In other words, how they are functionally connected. For the rest of the talk, I'm going to use those terms interchangeably just for simplicity. I'll talk about functional connectivity. And by that, I don't mean actual structural connections in the brain for aficionados who may be listening. Rather, I mean how these different brain regions are correlated over time, which we think is a proxy for how brain regions are connected with one another. Now, what you see here is a map of how each region in the brain is connected with every other region. And that gave us a tremendous amount of information. So there are something like 33,000 unique functional connectivity features in this map. And we were pretty confident that most of these would have nothing to do with depression. The brain isn't globally changed necessarily by depression. Rather, most of what we know about depression studies from the last few decades suggest that particular areas of the brain are altered. And so we knew that in order to accomplish this goal of subtyping patients, we would need a way of selecting which of these connectivity features are most important in depression and clustering patients on that basis. And that's what you see depicted here. For aficionados or technical experts who may be listening, the method we used here is called canonical correlation analysis, and it's really actually simpler than it sounds. The idea is just that we look for different combinations of connectivity features that are correlated with predictive of different kinds of clinical symptoms. And what we found were at least 2 factors, or that we'll call it for lack of a better term, 2 factors. One, you see depicted here a set of brain regions. Connectivity between these brain regions was predictive of the degree to which patients reported feeling anhedonic, anhedonia referring to a loss of interest in formerly pleasurable or enjoyable activities. So connectivity in these brain regions was predictive of anhedonia in our patients, and to a lesser extent of psychomotor slowing, feeling kind of lethargic, not able to get out of bed in the morning, and guilty rumination. And then we had a second set of connectivity features, the distribution of which you see depicted here, and they were not predictive of anhedonia or psychomotor slowing. Rather, they were more strongly predictive of anxiety and insomnia. And using these kind of connectivity factors, we then clustered patients. And what you see here are our initial results of that clustering. this is a dendrogram, and the main take-home message here, again, it's simpler than it looks. It's just that there are at least 4 groups of patients. In this dendrogram, the linkages, these lines kind of like in a family tree that connect branches of the tree, the height of the linkages is proportional to how different are the things being clustered. And what you can kind of appreciate is that the links between clusters are much higher than the average link between a patient with between 2 patients within a cluster. In other words, these clusters are defining groups, subgroups of patients with depression who have relatively similar patterns of abnormal connectivity in the brain, similar within a cluster, but different between clusters, and that's kind of the important message here. And before I proceed and tell you a little bit more about what I think will be most interesting to patients and family members, I want to emphasize that we don't think that this is the final or even the best solution to this problem of subtyping in depression. We think that the subtypes that we discovered were driven in part by the data that we had available to us. But what I want to try to convince you of is that this is just one solution, and that this solution is potentially useful for rethinking the way we go about diagnosing patients with depression, and ultimately for mapping treatments to those individual patients. So let me give you a couple of examples of that. Again, this is kind of a complicated slide, but the take-home message is simpler than it looks. What this shows you is a map of what's abnormal in the brains of people with depression in terms of how different brain regions are connected. Here, cool colors like you see here depict brain regions whose connectivity is reduced in depression, and warm colors depict brain regions whose connectivity is increased in depression. And this map depicts connections that were abnormal, that were altered in all of our patients with depression. They were shared across all the 4 subtypes. And what we found was that the degree to which connectivity in these circuits, these networks, the degree to which connectivity was altered was predictive of these symptoms that pretty much all depressed patients had. And they included depressed mood, anhedonia, and low energy and fatigue. In our data sets, about 97% of all of our patients had at least mild levels of these symptoms. And patients with more altered connectivity had more severe depressed mood, anhedonia, and low energy. So what we think this tells us is that there are certain connectivity features that all subtypes, all 4 subtypes share, and they're predictive of symptoms that most patients with depression have. But at the same time, there are other connectivity features that are unique to the subtypes, and that's what you see here. There are 4 maps. Again, cool colors means reduced in depression. Warm colors mean increased strength in depression. And what you can see just by glancing at these 4 maps corresponding to the 4 subtypes, you can see just by glancing at them that they're very different. That DSM assigns a single diagnostic label, major depressive disorder, to at least 4 groups of people with very different patterns of altered connectivity in the brain. And interestingly, these altered connectivity patterns were also predictive in kind of intuitively appealing ways, and we need to test this going forward, but they were predictive in intuitively appealing ways of differences in clinical symptoms. So here's a few examples. Patients with subtypes 3 and 4 had elevated levels of anhedonia, that is a loss of interest in previously enjoyable activities, compared to patients in subtypes 1 and 2, whereas patients in subtypes 1 and 4 had elevated levels of anxiety compared to 2 and 3. There were also differences in insomnia, energy levels, and psychomotor retardation or psychomotor slowing. And that was quite interesting to us as well, for reasons that I will touch on again in a moment. Before proceeding to tell you a little bit about how we think we can use this approach to subtyping to improve the way we map treatments to individual patients, I want to tell you a little bit about how we think this approach might be useful for developing new biomarkers. So biomarkers biomarkers are just biological measures that we can use to help diagnose an illness or to help guide treatments for that illness. And in many areas of medicine, biomarkers have kind of transformed the way doctors think about helping their patients by mapping individual treatments to the individual patients who are most likely to benefit from them. But in psychiatry, biomarker development has been difficult for all the reasons I just described, particularly related to the challenges of weak correspondence between our diagnostic labels and the underlying biology of the illness. So what we wanted to do was test whether we could use this approach to subtyping to develop brain scan-based biomarkers for diagnosing these subtypes. And again, this is a complicated slide, but the message is relatively simple. What you can see is that we optimize different steps for diagnosing these subtypes based on the brain scans. And at the beginning, the performance of our biomarkers was poor. It was barely better than chance in differentiating healthy control subjects and patients. But with a lot of optimization, we could improve the performance of those biomarkers to a little short of 90%. So we think that's maybe not quite what we might want for use in a clinic, but it's getting close. This gives you a little clearer sense of how well these biomarkers performed in the individual subtypes. Again, you can see in the range of 84 to 91% correct diagnoses for differentiating depressed patients and healthy controls. Next, we wanted to ask a related question. Having developed these biomarkers, these basically statistical classifiers for diagnosing the subtypes in individual patients, we wanted to ask whether they were stable over time. And that I think is a really important question because it provided kind of a sanity check for us that what we were doing made sense. We wanted to make sure that we weren't clustering subjects on the basis of something that didn't really mean anything for depression. Like, for example, you might imagine that if a patient who participated in our study had a fight with his wife, an argument with his wife before he came into the scanner, or if that patient didn't sleep well last night, or even depending on what the patient was thinking about when the patient was sitting in the scanner, all of these factors might influence the brain scan that we obtained from that patient. It might influence the measure of connectivity that we were obtaining using those brain scans in a way that has nothing to do with depression. So we wanted to make sure that we weren't subtyping people on the basis of something that had nothing to do with depression. And to do that, we asked a really simple question, and that's whether if a patient gets diagnosed with subtype 1 today, and we rescan them 5 weeks later and they're still depressed, will they still get diagnosed with subtype 1 at that second time point? If these subtypes are kind of clinically, biologically meaningful subtypes, then you'd predict that they would. And that's indeed what we found. Most people who were diagnosed with subtype 1 at scan 1 had the same subtype diagnosis in the second scan 4 to 5 weeks later. And so on for subtypes 2, 3, and 4. So the fact that these subtypes were stable over time was really helpful in telling us that perhaps we were onto something in terms of these subtypes being clinically meaningful. And then lastly, I told you a little bit about that second data set that we used for replication purposes, and this is also really important. Again, if there's any aficionados of these methods listening to this talk, they will know that these methods, what we call machine learning methods, are kind of notorious for overfitting to idiosyncrasies in the data that they're trained on. In other words, the biomarkers might work really well in the data set that we train them on, but then when you test them on a new data set from new subjects who were scanned in different cities on different brain scanners, they might not perform well at all. So we wanted to make sure they replicated well, and indeed we found that they did. That we could correctly diagnose individual patients with depression and differentiate them from healthy control subjects with accuracy rates in the range of 80 to 92%, roughly similar to what we found in the training sample. So again, that was kind of reassuring to us that we were on to something, and that this was likely to be meaningful. Okay, in the last couple of minutes, I'm going to shift gears, and I'm going to tell you about two last kind of quite interesting sets of findings. One of them has to do with how these subtypes cross conventional diagnostic boundaries. So what I've shown you so far tells us that DSM, our current diagnostic system, assigns a signal label, major depressive disorder, to people with at least 4 very different patterns of altered connectivity as measured by these brain scans. What we wanted to ask next was kind of the converse, and that's whether DSM assigns different labels to patients who have related diagnoses in terms of their brain scans. And the diagnosis that we focused on for this purpose was generalized anxiety disorder. And we did that for a number of reasons. We know that generalized anxiety disorder and depression tend to run in families together. We think that the genetics that contribute to risk for depression are probably related to the genetic risk factors for anxiety. We know that many kids who are anxious grow up to be depressed. We know that many adults who are depressed also have symptoms of anxiety. So there was a lot of reason to think that these two diagnoses were related. So we just asked a couple of simple questions about how our classifiers, our biomarkers, treated patients with generalized anxiety disorder who did not have a diagnosis of depression. And what we found here, again, this is a map of what's altered in the brains of people with generalized anxiety disorder. Warm colors mean increased in anxiety compared to healthy controls. Cool colors mean decreased. And what you can see in this little Venn diagram is that although the connectivity features that define depression are different from the connectivity features that define anxiety, there's substantial overlap. About 22% of the connectivity features that define generalized anxiety disorder are also altered in depression. And when we asked how our biomarkers for the 4 depression subtypes treated people with generalized anxiety, what we found was quite interesting. We found that that a majority of people who had a diagnosis of generalized anxiety disorder tested positive for one of our subtypes. So about 31% were classified as not depressed, but the remaining tested positive for one of our subtypes, and the vast majority of those tested positive for subtypes 1 in blue or subtype 4 in black. And you'll recall from the data I showed you a few minutes ago that the 2 subtypes in our depression data set that had elevated levels of anxiety were subtypes 1 and subtypes 4, the same subtypes that many of these anxiety disorder patients are being diagnosed with. Furthermore, when we look at other symptoms, an interesting pattern emerges, and that's that their anxiety symptoms in these generalized anxiety disorder patients, they do not vary by subtype, and that kind of makes sense. All of these patients are anxious. They have high levels of anxiety, as indexed by this self-report. But their depressive symptoms did vary by subtype. That is, patients who were diagnosed with one of our subtypes had significantly elevated depressive symptoms compared to the healthy control, the patients who had anxiety but were diagnosed as not depressed. And furthermore, the patients who were diagnosed as subtypes 3 or 4 had elevated levels of anhedonia compared to subtype 1, which was in turn increased compared to people who were diagnosed as not depressed at all. So what we think this shows is that these subtypes that we've identified based on connectivity measures in the brain cross conventional diagnostic boundaries in interesting and intuitive ways, and there's some consistency in how their symptoms, their depressive symptoms, their anhedonia symptoms, how they relate to their brain scan diagnoses. But there was one thing that was bothering us a little, and that's whether perhaps the connectivity measures that we were detecting with these biomarkers were kind of non-specific for any kind of generalized form of psychopathology. In other words, does anyone with any kind of mental illness tend to test positive for one of our subtypes? And what we found by looking at patients with schizophrenia was that that was not the case. So in this sample of about 75 patients with schizophrenia but not depression, almost all of those patients tested negative. In other words, these diagnostic biomarkers for subtypes of depression, they cross diagnostic boundaries, conventional diagnostic boundaries, in interesting and intuitive ways, but they still retain some level of specificity, which is important if we're ever going to be able to use an approach like this for thinking about how we go about treating patients. And of course, that's what matters most. Lastly, in just the last few minutes before we kind of take a break for questions, I want to tell you a little bit about what's maybe most exciting, and that's how we think we can use this approach to improve the way we go about treating patients. The treatment that we focused on here is called transcranial magnetic stimulation, TMS. For those of you who aren't familiar with it, TMS is an established FDA-approved treatment for depression. It's non-invasive, but it's a brain stimulation technique, and the way it works is by applying a magnetic field, a rapidly fluctuating magnetic field, to the brain through the scalp. So it's totally non-invasive. A person just comes into a psychiatrist's office, and it can be administered right there in the office, and the patient goes home right after the session. And we've known for many years that when you apply this treatment, TMS, to the brain repeatedly over a period of several weeks, that patients with even treatment-resistant depression who have not responded well to other antidepressants, they tend to get better over time. And importantly, we've also known from our own work and from other folks' work as well that brain measures of connectivity, like the ones I was showing you before, are sensitive to TMS. So TMS alters them. What you see here is kind of pre-TMS and post-TMS. And the connectivity features that are altered in depression get normalized after TMS in patients who are undergoing treatment. And furthermore, the degree to which a person has abnormal connectivity or altered connectivity in some of these networks before they get TMS is predictive of their likelihood of going on to respond to TMS. And that's important because it tells us that there's maybe some potential for using these brain scans for informing which patients are most likely to benefit from that treatment. And that's really critical because we also know that this treatment, while it is effective, it doesn't work for everyone. And like many other antidepressants, it takes 4 to 5 weeks to have any effect. And of course, that's really frustrating for patients and their families and for doctors as well when they try this treatment only to learn 5 weeks later that it's really not working and they need to kind of start from scratch and try something else. So what we wanted to test was whether we could use our approach to subtyping patients with depression to inform these kinds of decisions about who would be most likely to benefit from TMS and who wouldn't. And what you see here are the initial results of that experiment, that test. What we found was, so we obtained brain scans. This is in collaboration, by the way, with my colleague Jonathan Downer at the University of Toronto. We obtained brain scans from patients with depression. We subtyped them, and then these patients received TMS targeting a brain region called the dorsomedial prefrontal cortex. It basically lies along the midline of your head, kind of at the front of the head. And this brain region we know is responsive to TMS and can be used for treating patients with depression. And what we found was that patients who were diagnosed with subtypes 1 or subtype 3 were much more likely to respond to TMS than patients who were diagnosed with subtype 2 or subtype 4. Here you see another way of depicting that same data. The degree to which a person improved was much larger in subtype 1 and to a lesser extent in subtype 3 than it was in subtypes 2 or 4. These people in subtype 1 in particular showed really strong response rates to this kind of TMS. And what you see here is kind of a second step towards what we think of as potentially precision medicine in psychiatry. Here we asked whether we could design a prognostic biomarker, a statistical classifier that could be used for predicting who would respond to TMS and who wouldn't, based on different kinds of measures that we could obtain from those people prior to treatment. What you see here are the results for predicting responders R and non-responders NR, based just on brain connectivity measures as indexed by that fMRI brain scan I described earlier. And what you can see is that we can do okay, about 78, 79% correct prediction rates, but that's still not as well as we'd like. To actually start using this in clinical practice. But you can see by adding in the subtype diagnosis, we can improve the performance of those biomarkers significantly to something in the range of 87 to 94% correct predictions. And the last thing we wanted to test was whether we could just use clinical symptoms to do just as well, because you'll recall I told you before that, that our 4 subtypes had different patterns of clinical symptoms. And what we found was that although clinical symptoms like the degree to which someone was anhedonic or anxious or not sleeping, they were predictive to some degree of whether someone would go on to respond to TMS or not, they weren't nearly as predictive as a combination of these connectivity features and their subtype diagnosis. So we think this is promising. I want to emphasize, I always emphasize this when I'm giving this talk to patients and families, that I think we're really excited about this work, but it's still early days. It's really critical that we validate this, that we replicate these findings, and that other groups replicate them as well, and test to what extent these methods can really be used for predicting individual treatment response rates in individual patients and guiding treatment decisions on a more widespread basis, not just kind of in the relatively unique situation of a patient participating in a research study at an academic institution. So we don't think that these are ready for widespread clinical use yet. That's still some ways away, but we think what this data tells us is that this approach holds a lot of promise, and we're pretty confident that we can improve upon it further, and that other investigators will probably figure out other ways of doing this even more effectively. And with that, before we stop to take some questions, I just want to highlight some of the folks who were responsible for carrying out this work. In particular, Andrew Drysdale, a very talented MD PhD student who is now a psychiatry resident at Washington University. And the folks highlighted in red here: Mark Dubin, Faith Gunning, George Alexopoulos, Jonathan Downer, BJ Casey, Mike Fox, Alvaro Pasqualeone, Helen Mayberg, Amit Etkin, and Ellen Schatzberg, who were generous enough to share their data with us and make this work possible. And of course, none of this would have happened without generous support from the Brain and Behavior Research Foundation and our other sources of financial support. These are all academic institutions. I should have mentioned I don't have any conflicts of interest or any other financial stakes in this work. So I think with that, I'll pause. Do we have some time, Jeff, for questions?
Yes, we do. First of all, Conor, thank you so much, first of all, for the work that you've been doing on this, and for really explaining some very complicated issues in a way that the general public, the lay public, can understand. So I think this is very exciting. And as you said, it's not quite ready for prime time for individual patients, but has the potential to get to that point. And one of the questions that a number of people asked is, what will it take to get to the point that it can be used for actual patients in clinical care?
Yeah, I think that's a really critical question. It's one we think about here in my lab every day. I could talk about that ad nauseum, and I won't, but I'll tell you just 2 points that I think are really important questions we need to answer before we bring this into widespread clinical use. First of all, this work needs to be replicated. I'd like to see other groups using similar methods and testing whether it works in other people's hands at other academic institutions and also outside of academic institutions, because oftentimes the kinds of patients that participate in research studies may not necessarily be representative of the kinds of patients that we see in the broader world. And sometimes that happens in ways that are hard to predict. We try to control for that, of course, but it's really critical before we start using these kind of methods all over the place that we know how well they work and that we know under what circumstances they don't work so well. What are the limits of how they work and how they don't work? So replication and testing by other labs, I think, is a really critical step. A second step that I think will be really important is testing whether a similar approach can be used for predicting response to treatments that are in more widespread clinical use today. So we focused on TMS. The brain region, I didn't mention this, the brain region that we were targeting is the dorsomedial prefrontal cortex, which is actually not the FDA-approved site. It is one that we know works from multiple published studies, and we focus on that one for a number of reasons that I won't get into here. But we really need to test whether a similar approach also works for the FDA-approved dorsolateral prefrontal site, and maybe even more importantly, whether this approach can be used to predict response to medications, first-line therapies for depression like fluoxetine, escitalopram, sertraline, medications that probably a lot of our listeners today are more familiar with than TMS. That will also be really important.
Right. So really the goal is to be able to use this technique to then help the physician decide with the patient what medicine or other treatment is most likely to help them. Talk therapy could be one of those options as well in terms of treatment for people.
That's exactly right, and I'd like to actually, I'm glad you brought that up. I'd like to highlight a recent study from one of our collaborators, also a brain and behavior research Foundation scientist, Helen Mayberg. She's done amazing work showing that we can use this kind of brain scan approach to predict who's most likely to respond to a drug versus who's most likely to respond to psychotherapy. This question of differential treatment response prediction, I think that's really critical, and that's something we'd like to test maybe in collaboration with Helen at some point in the future as well.
Yeah, very useful and important, as you pointed out, given the time it takes for medicine or other treatments to work. If you have a better chance of it actually working first time around, it makes a big difference in people's lives.
Right. That's right.
One question that a person asked relates to bipolar disorder. You spoke about general anxiety. You spoke about comparing it also to people with schizophrenia. Any information about bipolar disorder, depression and bipolar disorder, and what you're seeing with these types of studies?
Yeah, that's a great question. Again, another one that we think about a lot in the lab. I think it really gets to the root of a problem, which is that, for the most part, we don't need brain scans to diagnose depression. You can see it in a friend or family member without any special training a lot of the time. But one diagnostic question that can be really hard and that has really important clinical implications is distinguishing between unipolar depression and bipolar depression. For listeners who aren't aware of the difference, bipolar depression refers to a kind of depression that happens in some patients who also experience periods of remarkably elevated mood and these patients tend to respond differently to medications. Some of the medications that are useful for unipolar depression can actually be harmful for bipolar depression. So we'd love it if this approach to subtyping patients could differentiate between bipolar and unipolar. Unfortunately, we don't know the answer to that yet. All of the patients in our study that I just described had unipolar depression. That's an important limitation. We are working to test whether a similar approach could be useful for differentiating bipolar patients, but that's going to mean analyzing a different kind of data set, one that we haven't fully analyzed yet. So I don't know the answer to that, but hopefully if you check back in a year or so, maybe we'll have more information on that.
Well, we may need to bring you back to give the next webinar on that, because obviously it's very important. Another important issue in depression and other psychiatric conditions is the issue of suicide risk. And I'm curious, is there anything in your data and your analysis that can be helpful in terms of potential biomarkers for people who might be at greater risk of attempting suicide?
Yeah, that's a really important question too. Suicide obviously is probably the gravest outcome that we face as psychiatrists and mental health care providers in treating our patients. It's one with really huge public health implications. And a challenge in studying suicide is that it's thankfully it's a relatively rare event. And so we don't know as much about, like most of the patients in our study, for example, don't have a history of suicide attempts. And certainly there were no completed suicides or suicide attempts during the duration of our study in any of the subjects that were involved. What we do know is that some of the questionnaires that are administered, the clinician rating scales and the self-reports, ask patients and their doctors about suicidal thinking, which is not the same as suicide attempts. And it does look like provisionally that one of these subtypes is associated with an increased risk of suicidal thinking, but we really need more data to say anything conclusive about how this measure of risk for suicidal thinking relates to people's risk for actual suicide attempts, which is, of course, what we care about more. And we think that the way to tackle this is by collaborating with investigators who have much larger scale data sets and information to speak to that question directly, and we hope to be able to address that soon.
Obviously, a very important issue. You spoke about the general anxiety group, and you mentioned the important point that often people at a younger age with anxiety may be at higher risk of developing depression. Do you have any information on looking at people who have not yet developed depression, but that may be at higher risk of developing depression, and therefore there may be methods of prevention that can be done before depression really takes hold?
Yes, that's a great question. Again, another one that we're really interested in. It raises a related issue, which is, whether these subtypes, which we're diagnosing on the basis of altered connectivity in a brain scan, whether that altered connectivity is something that precedes the onset of symptoms. Maybe it's there before a person ever gets depressed. Maybe it's kind of a stable feature of that person's brain that renders them kind of at risk of becoming depressed. On the other hand, it might be the case that these altered connectivity features are kind of a state-dependent feature of the person's brain that varies with their mood state. So it's kind of indicative of the fact that the person is currently depressed, but if you rescan them again a few months later when they were no longer depressed, you wouldn't see these changes. although we don't have data to address the question of risk for developing depression in patients who've never been depressed before, we do know that if we look at patients repeatedly over time, it's probably the case that what we're measuring here is a combination of those two things. Some of those altered connectivity features are stable features of the person's brain, and some of them vary with the person's mood state. The fact that some are stable features of the person's brains suggests to me that they might precede the onset of symptoms. They might be kind of a sign of risk for developing depression, in which case, like if we scanned adolescents or people in young adults who are at high risk for becoming depressed, they might be predictive of depressive outcomes. And, that itself could be really powerful because there are lots of interventions you might imagine deploying that don't involve medications or TMS or brain stimulation, easier interventions like stress reduction and just knowing who's at risk and being able to warn those people to avoid other risk factors for depression. That could be really powerful. We're working with collaborators who have data that might help us address that, but again, we just don't know the answers to those questions yet.
Great. Well, while you've developed a lot of very important information, in some ways all of these new developments bring up new questions that need to be looked at as well. So it's a very exciting time. As a last question, I want to ask you, what do you see down the road? What do you envision in 3 years, 5 years, 10 years down the road in terms of how this is going to affect clinical care and people's lives?
Yeah, that's something I think about all the time too. I think in the near term, what I'm most excited about is using this approach. And again, I don't think that it's going to be just my lab doing this. I think there are labs all over the country who are using similar methods to tackle related problems. Using this approach to guide the selection of existing treatments, I think, is really powerful. I think what I'm most excited about in kind of the near term is an approach similar to what Helen Mayberg has done, which is differential treatment response prediction. That is, can I tell someone not just that this treatment is or is not going to work for you, but rather I can tell them treatment A is not going to work for you, but treatment B is going to work for you. And that would be really powerful, and I think it could do a lot to kind of bend the curve on those statistics that I described at the beginning of the talk today about the kind of accumulating burden of mental illness over time that's due in part to the kind of frustrating trial and error approach to treatment that we're often forced to use. And then the longer term, I think what's equally and maybe even more exciting is the potential for using this approach to think about developing fundamentally new treatments. Many of the treatments we use today are really closely related to one another, and it's been frustrating for many scientists and doctors and patients that we haven't seen fundamentally new treatments for depression in some time now. There are some promising candidates on the horizon, but I think that rethinking how we diagnose mental illness and depression in particular, and identifying kind of more homogeneous subtypes might make it easier to understand the biology of depression, and if we understand biology, then hopefully we can develop kind of fundamentally new drugs and other interventions that aren't just kind of cousins of the drugs that we have today.
Well, Conor, I think that is a vision we all would love to see. And I want to, again, thank you for the work that you're doing and for joining us today to share this exciting work that you're doing in your lab. I want to also thank everybody who's joined us. All of the research that we fund is made possible through private donations. So please consider making a contribution by visiting our website, bbrfoundation.org, or call 1-800-829-8289. This webinar has been recorded, so if you've missed any portion or would like to share it with a family member or friend, please visit the events and webinar page on our website. And I hope you'll join us again next month when Dr. Holly Schwartz, professor of psychiatry at the University of Pittsburgh, will present psychosocial interventions for maternal depression, impact on school-aged children. This will take place on Tuesday, October 10th at 2 p.m. Eastern Time. Thank you again for joining us, and enjoy the rest of the day. Take care.